others. I really enjoyed observing nature. I also enjoyed writing. And I enjoyed explaining about what I observed to my friends. So it's actually not a surprise that later on I became a researcher and a professor, because this is what I do now on a daily basis. I do research, I write, and I teach. I was fascinated by observing insects. For example, I collected ladybugs and I observed their behavior. And it really passionated me to learn more about biology. And this is how I started studying bioengineering sciences. And I became very inspired by two professors. Professor Glansdorff, who introduced me to the fascinating world of microbes, and Professor Charlier, who became my mentor. I learned that there's a huge, invisible world of microbes out there, on a micrometer scale, and that there is a huge diversity in the microbial world. Microbes are indeed everywhere. They thrive in every imaginable habitat on Earth. Whether it's in your guts and on your body, in the soil, in the ocean, and even in the most extreme environments where you would not expect that life is possible there. And it's these extreme environments that are absolutely fascinating. And I learned from my mentors that there's a lot of microorganisms living in these environments. You can find a lot of such environments in Yellowstone National Park. Journey. I wanted to understand much more about these microbes that are living in these hostile environments. And here you can see me with, in the background, my mentor, Professor Charlier, visiting just such an habitat in the Azores in Portugal. I wanted to understand how microbes have adapted to thrive in conditions of extreme temperature up to 80 degrees, or extreme pH as low as pH 2. You can compare this with almost boiling citric acid. How they can not only survive, but also grow in these conditions. And I wanted to understand how evolution has shaped the success of a microorganism in such a specific habitat. And there's two important elements in explaining this. The first one is time. If we consider the evolutionary history of life on Earth, and we go back in time to the origin of life, which has happened on, let's say, January 1st, it's only until November 6th that life on Earth was only exclusively microbial, and on November 6th, multicellular life has originated. November 20th, plants have originated, and only on the last day of the year, December 31st, we as humans have originated. But this means that microbes have had so much more time to evolve and to adapt. And moreover, they evolve much quicker than most multicellular life forms. So this is already an important reason. The second reason is that, maybe you're not aware of this, bacterial cells are continuously changing their genes, their genetic makeup. They are exchanging DNA with each other. Here you can see two bacterial cells that are exchanging DNA through a mechanism called conjugation. They can also take up DNA from their environment, and they can make modifications in their own DNA. And they have very sophisticated mechanisms to do this. One of those mechanisms you might have heard of is CRISPR, which we have harnessed as scientists to use for genome editing of other organisms. So this means that bacteria are continuously, or microorganisms are continuously changing and creating diversity in their genetic makeup. And taking those two factors together, this long time and these efficient mechanisms of genetic diversity has led to a huge microbial diversity on Earth. We can see in green here the eukaryotes, with here humans and animals, and here plants. And the rest of this phylogenetic tree is only composed of microbial species. So there is a huge diversity of microbes on Earth. And billions of years have enabled the selection of the fittest and best adapted genotypes to thrive in specific habitats, in all habitats imaginable on Earth. And this time scale of billions of years is important to remember. We'll come back to this. During my PhD, it was the start of the genome sequencing era. This was really amazing to me. And I learned firsthand from my mentor, Professor Charlier, how we can sequence microbes and how we can learn the code of the properties of the phenotype that we observe. So it's an old classical biochemical method with which we can read DNA. In 2007, I defended my PhD, and I'd spent several years to unravel the genetic mechanisms underlying gene regulation in microbes thriving in these extreme habitats. You can imagine that one microbe is encoded by 1 to about 10 million base pairs, and we have tried to unravel this code, not only learning about genes, which encode the structural properties of a cell or the enzymatic properties, but also about codes that determine how the microbes can respond to changing conditions in their environment and how they can regulate gene expression. And this is what makes them so successful in habitats, what makes them robust. These kind of sequences would consist of promoters, ribosome binding sites, regulators, operators, terminators, and you have a very complex circuitry which reflects the complexity of biological systems. I further continued studying microbes and mechanisms of gene regulation during my postdoc years until in 2014 I had the opportunity to start my own group. And at that time I realized that I wanted to take this knowledge a step further, and I also felt the pressure and the urgency to try to valorize this knowledge, to find solutions for challenges that we are facing as a society, as humanity. Because we started to realize more and more that Earth is changing faster than ever, and it's due to human impacts. It's clear that we are living in a new geological era, the Anthropocene, which is viewed as the period during which human activity has a dominant influence on climate and our living environments. How can microorganisms help us to find solutions? How can they offer an alternative for unsustainable industrial models? And this is what we are facing now, because the current industrial model consists of a linear economy, where we start from fossil resources that are depleted and that are finite, and where petrochemical industry converts this into fuels and chemicals, while during this conversion CO2 emissions are generated, and also at the end of the lifespan of these products, think about plastics, they can create a burden on the environment because they cannot be degraded and are polluting our living environment. But we know that microorganisms can also perform chemical reactions. Think about yeast. We are using yeast fermentations for a long time, for example, to convert sugars in alcohol, like the cocktail you are drinking. So we can actually consider microorganisms as small chemical factories. And in the case of yeast fermentations, we as humans have been selecting good producers for centuries so that these fermentation processes were optimized. We were doing this just with naturally occurring genetic variants. And we can look here at the time scale. It took us centuries to select for those good producers. But besides alcohol, there's so much more that we can produce with microbes. They can actually, with all this diversity that they evolved throughout those billions of years, they can convert virtually any chemical compounds in another chemical compound. I was amazed to learn that there's a lot of bacteria, and you see here bacterial cells, that produce plastics, bioplastics. Up to 80% of such a cell is composed of a material that resembles the plastic that we know out of our daily life. But this plastic is produced in a sustainable way. It's produced from a biomass feedstock instead of from fossil resources. And the bioplastic itself is biodegradable, so it will not cause any negative impact in the environment. This is our linear economic model that I was just explaining, where we see the CO2 emissions coming from the petrochemical industry, and then this non-processable waste that is produced and pollutes the environment. With microbes, we can offer an alternative. And virtually any chemical compound can be converted into another compound. We can start from biomass feedstocks like agricultural side streams that are not used anymore, or food waste, or brewery side streams, or seaweed, or even CO2 itself could be used as a starting point. And microbes can convert this into a diversity of different products that we can use in our daily life. Feed and food additives, medicines, cosmetics, detergents, bioplastics, biofuels, or biomaterials. And this is a much more sustainable model that is not linear anymore, by starting from plant biomass that has captured CO2 from the atmosphere during the growth of the plants, converting it into biofuels or bioplastics that can then also be more biodegradable, leads us to an almost or usually CO2 neutral process. So it's a very important alternative to our current or previous linear economic model. In 2017, I also encountered a new type of microbes that are just as fascinating as bacteria, namely filamentous fungi. You might know filamentous fungi from in forests by encountering their fruiting bodies, mushrooms or fruiting bodies that you can see growing on dead wood. But this is only a part of the structure of these organisms. The vegetative part of the organism grows below the surface in the soil under the form of so-called mycelium. These are tubular cells, which we also name hyphae, with very strong cell wall structures. And when they grow, they actually form a material by itself. And they can grow by making use of a diversity of organic compounds. They can also grow on lignocellulosic feedstocks. This is what it looks like in the lab. We can take any kind of fibers or organic substrate, sterilize that and put it in a mold, and then inoculate it with our organism, with the filamentous fungi. We let it grow for about 2 weeks, and the mycelium forms this white color, and it glues the fibers together. After a drying process in which we kill the fungus, we obtain a very interesting lightweight material. It resembles isolation material or some kind of material that we could use to construct a house. We can further process it in different ways by pressing it, heat pressing it, so that we obtain materials with a diversity of properties. And these materials, which we call mycelium materials, are an example of a material that is produced in a completely circular concept. So we start from plant material as a substrate, we sterilize it, inoculate it with the organism, which you can do by starting from mushrooms, place it in a mold and incubate it at a certain temperature and humidity for about 2 weeks, after which it is dried in the oven, processed and used. And at the end of the lifespan of the material, when you don't need the product anymore, you can just put it in the soil, put it in your garden, and it will decompose, and it can be used as a substrate to grow a new material. These materials don't have any negative impact on our environment, not during their production, not at the end of life. And there's a whole industry that is emerging and that is developing applications with these mycelium materials. You can think of acoustic panels or floor tiles. And you can take it a step further. While in the previous example we were looking at composite materials that are not only composed of the organism, the fungus itself, but also of the substrates, the fibers. We can also grow the organism in very controlled conditions in the lab, so that we obtain a layer of pure fungal biomass either on a solid state fermentation or a liquid state using liquid substrates. You can easily harvest this layer of mycelium, and it has very interesting properties. If you have the chance of feeling it after the presentation today, I invite you to do so. It feels a bit like velvet, and the interesting or the fascinating aspect is that we are now looking at a material that is 100% composed of a microbe. So it means that the genetic makeup of this organism determines the material properties, and we can start tuning it by changing the environmental conditions or by tuning the genetics of the organism. And also during the growth process, because it's a biological system, it's regenerative, we can actually envisage that we can shape and tune the material. For example, by playing with airflow. The shoe was formed during this growth process. And actually Adidas is working on its first prototype with mycelium materials. I'm curious, would you be willing to wear shoes that are made out of fungi? Maybe you can raise your hand if you would be okay with that. I'm happy to see this response. That's great. We should also communicate this to the companies who are developing these prototypes. Because it's an important example of a sustainable consumer goods. I think I convinced you. These new products, we need them as a replacement for products that are putting such a burden on our climate and our living environment. And the technologies exist. We have already been doing a lot of research, and we know already what is possible with microbes, both bacteria and fungi. But still, it's really hard. And it's not yet put in practice. So what is preventing us? We know that it's urgent to make this transition, so why is it not happening? What's preventing us from transitioning to a bioeconomy? And the answer is the costs. Currently, the cost efficiency of industrial biotechnological processes is too low to convince or to enable the industry to make the transition. For the same cost, for the same price, you can produce much more products, for example ethanol, which is a biofuel, in a classical petrochemical conversion and taking into account the current cost of fossil resources, fossil oils, as compared to a microbial conversion in a bioreactor. And this is really a bottleneck that we need to overcome. A possible problem that explains this bottleneck is that the microbes that we use in the bioreactors are not optimally adapted to the bioreactor environment. And they are not evolved to produce high yields of our products. They did not have billions of years of evolution to adapt to this very anthropogenic environment. And we even did not have centuries, usually we did not have these centuries to select for the best producers working with natural strains. The problem is that it's urgent. We only have a few years to find a solution. And this solution might be offered by synthetic biology. So what is synthetic biology? And how is it different from the classical genetic engineering, which has been established already a long time ago? Synthetic biology means that we apply the engineering paradigm of systems design to biological systems in order to produce predictable and very robust systems with novel functionalities that do not exist in nature. And these functionalities could mean the production of a chemical or another product. We can do this by making use of the natural mechanisms for gene or genetic modification, such as CRISPR, for example. But what is different from classical genetic engineering is that with classical genetic engineering, we just took one gene and introduced it into a microbe. And this gene could, for example, bring the message to the microbe, convert sugars in ethanol. It worked a little bit, but not too much, because there was not really an alignment with the rest of the metabolism, the rest of the circuitry of the microbe. And I was telling you how I investigated during my PhD and postdoc years the complexity of mechanisms of gene regulation. Everything is balanced in the cell. And this new gene that was introduced was not fitted into that complex system. In synthetic biology, we take this complexity into account. We will not just introduce this gene, but we will also introduce all those other elements that will ensure a good regulation and an integration with the rest of the metabolism and its regulation of the microbe. So we're thinking about promoters and ribosome binding sites and a regulator, operator, terminator. All these sequence elements are also integrated. And by doing this, we bring a message to the microbe that says, convert sugars in ethanol at high yields and also in conditions of stress. And this could lead us to a much better producer, because we are actually sourcing natural mechanisms. So we could compare all of these sequence elements here with Lego bricks. And what we need to do, synthetic biology, is first of all a very thorough biological knowledge. This is also why synthetic biology has only emerged quite recently as a new scientific domain. We need to have good knowledge of the naturally existing sequence elements, promoters, ribosome binding sites, genes, operators, terminators, regulators. And we need to have well-characterized, predictable, standardized, modular and orthogonal biological parts. This means that these sequences will function like we think they will, and that they will do this in a predictable way, independent from the metabolism and the regulation in the microbe itself. The second thing that we needed for synthetic biology to emerge is that we should not only be able to read DNA and to know all of the sequences that are present in natural microbes, but that we also need to be able to write DNA. We need to be able to synthesize long pieces of DNA that we can then integrate into the microbe. Bringing in a single gene of a few hundred base pairs or maybe 1,000 base pairs was still feasible with previous technologies, but now we're looking at entire circuits of different sequences. And this is what also stimulated the emergence of synthetic biology.
What if you could use this space to write 9,600 genes? Using the same footprint as a traditional 96-well plate, Twist Bioscience starts with a plain silicon wafer, like those used in the semiconductor industry. With microscale patterning, the resulting silicon chip is able to write over 1 million oligonucleotide sequences to generate nearly 10,000 genes at a time. Using conventional chemistry approaches on a miniaturized scale, we write DNA base by base. The DNA is then assembled into genes or other products smaller and faster.
It's not only needed to be able to synthesize these long pieces of DNA because we are looking at longer genetic circuits, but also for another reason. And this is a very important reason. Namely, it's sometimes misconceived by the audience that synthetic biologists can play God and that they know exactly what to add or what to change in a biological system, but that's not the case. As scientists, we are very humble and we know already a bit more about biological systems, but not everything. We cannot understand everything. And because of this lack of understanding, we need to try many different combinations of our Lego bricks. It's impossible to predict which one will work best and which one will lead to our very good producer that we need. And if we would want to try them one by one, it would take us again way too long, and we don't have that time. So the third important technology that led to synthetic biology emerging as a scientific field, as a domain, was techniques of high throughput screening. This allows us to make these large combinatorial libraries of different combinations and then to grow all those different microbes that we produce in a high throughput manner. For example, using a robotic platform so we can move quickly in an automated way. And we screen for the phenotype. So we use techniques so that we can quickly identify which one is the most successful combination. Okay, so we are talking here about genetically engineered bacteria. And in previous lectures, I sometimes had a few negative responses from the audience. So I'm again very curious. Are you afraid when I'm talking about genetically engineered bacteria? Do you think that these could be dangerous? Yeah? Yeah, I see quite some people. Thank you for being honest. Because it's a sentiment that is real and that is living with many people. From a science perspective, from a perspective as a scientist, I know that what we are doing here is the same as what is happening in nature. We're using the same mechanisms, and you can imagine that also in nature all combinations have already been tried because of these abundant mechanisms and this long time of evolution. But not all combinations still exist because only the fittest ones remain in the context of the natural habitat in which they live. However, I agree with you that we are moving very fast and that we should reflect on preventing these genetically engineered bacteria from spreading in ecosystems, because that could disturb the balance of ecosystems in unpredictable ways. So, as scientists, we are thinking about this. And as synthetic biologists, we are also thinking of developing in parallel so-called biocontainment strategies, which means that we can introduce also a circuit into the bacterial cell that says, commit suicide and being released outside of the bioreactor, so that we can contain it inside. I have a last question for you. Based on what I told you about synthetic biology, do you think this is bringing us further away from nature or closer to nature? Who thinks that it's bringing us further away from nature and making the distinction and the separation between mankind and the rest of nature even larger? A few people? Yeah. Is the other part of the audience thinking that it might bring us closer to nature? Yeah, I see quite some hands now. I want to illustrate this with an example. This was actually a real example. You might have heard of Ecover, which is a Flemish producer of ecological soaps and oils. And they actually make use of synthetic biology. They use synthetic biology to produce oils from which they produce soap. And they did this by creating engineered algae that are more performant in the production. And by doing this, and by relying on an industrial biotechnology process, they prevented the use of palm oil, which is typically used for the production of this kind of products. And so they prevent deforestation for the sourcing of palm oil. It's not a black and white story, and we'll need also other measures. And this kind of argumentation really depends also on the context, and it should be assessed also in each individual context. But I do believe that with synthetic biology, we can make use from bio-based processes and products much easier, and that this will help us to reduce the negative impact on the environment and the living conditions in which we live. And so this is a story of hope. I believe that by 2050 we will be living much closer to nature, and that partially this is with the help of microbes that help us to produce and consume in a very different way. Steve Jobs said, the biggest innovations of the 21st century will be at the intersection of biology and technology. A new era is beginning, and I think it's going to be a very exciting era with hope for the future. The second part of my story was not so personal, and that is because most of the concepts were developed and the research was done by a team of hardworking and talented scientists, my team at VUB, and I want to acknowledge them for all their hard work, and also, of course, the funders of this research. And I want to thank you for your attention